I will present you a little bit of what was done in Tropicsafe about the Italy. I will not tell you the history, it will take too long. So my presentation will be diversity of phytoplasmas associated with Grapevine Yellows in several Italian regions. And the author are Nicoletta Contaldo and myself, and a lot of our students and collaborators in Italy that are not listed here. As I mentioned in the previous presentation, and as you feel from the two presentations before me, the phytoplasma are different according with the different region of the world you are studying the Grapevine Yellows. The situation in Italy is that we know the presence of Grapevine Yellows since the 70s, but in the last 20 years, let's say, a growing number of diverse phytoplasmas were detected in the Italian vineyards. They were usually together with the for us historical phytoplasmas associated with Bois Noir and flavescence dorée. It's not the case that this disease has French names, because they were studied, and I need to mention here, for more than 50 years by Antoine Cauduel in France. He was let's say, the first one discovering these diseases before the discovery of phytoplasmas. He found the disease and he studied. As it was already told, the main vector of flavescence dorée is Scaphoideus titanus, and for the Bois Noir, the main vector known is the Alesius obsoletus. In the Tropicsafe, we focused on the three regions you see with star that are in the North Italy, Veneto, Emilia-Romagna, and Tuscany, where there is a flavescence dorée. We try to study the updated situation in some vineyards located in this area. For your information, flavescence dorée is just able to colonize until now, until middle of Italy. So Tuscany and Marche region are the southern limit of the disease. And then we studied the Bois Noir on other regions, mainly located on the east coast of Italy. Again, Tuscany, Marche, Abruzzo, and also Puglia region, together with some Emilia-Romagna. By verifying the situation, I will not speak about symptoms anymore. And this is the possibility to distinguish at least Bois Noir in blue and the two strains of flavescence dorée in red and green. Just using one 16S ribosomal gene and one enzyme, you see the three different profiles telling you have different phytoplasmas. But beside these phytoplasmas, it was possible to detect in some area other phytoplasmas, like the one you see in this slide, the 10B on Cabernet. 10B maybe is telling nothing to you, but it is the European stone fruit yellows, a phytoplasma disease that is mainly detected in plum and peaches. And also in Italy, you see in the last slide, we found 1B, that is the same group as the South African phytoplasma Johan Burger presented before. By studying further, we know now that they are not the same strain, but they belong to the same group. And what is increasingly found is that there is a lot of biodiversity in the phytoplasmas found in Grapevine in Italy. And this is very likely to be connected to the ecological variables that are influencing this kind of disease, insect transmitted, that are the management of the grapevine, the insect present that can be vector, and the phytoplasma strain. These are the main actors of this. As for the environment, I can just show you the difference we have in Italy. This is in the high mountain. You see in the circle the area in which you have a yellow symptom in Vineyard. This is just in a mountain. But you can have this. This was a result of testing in a flat area. And in one area, a few grapevine vineyard, we found the presence of group 5 and group 12, so flavescence dorée and Bois Noir, and also aster yellows. So what was found in South Africa and in Chile is not incidental. The presence of other phytoplasmas is also present in Italy. The project worked mainly in this way, considering the actor of this disease. We did several sampling experiments in several areas, honestly using the student performing the master thesis for this, And we collected the grapevine, the weeds around the grapevine, and the insect in the same area. And then the weeds were collected usually asymptomatic, just because they were in a vineyard showing symptoms. Some of the weeds we detected harboring phytoplasmas in common with grapevine, as Parthenocissus quinquefolia, you probably know, show also this kind of reddening symptom, and also Skinia japonica, reddening symptom. Both these plants were infected by phytoplasmas of stolbur, I mean, the same that are associated with Bois Noir disease. This is a list just to show you a bunch of other species we tested, and for some of them we found the presence of phytoplasma. This is a short list. We have a longer list that just to show what we found was the 16S R12A is a stolbur or Candidatus phytoplasma solani, or Bois Noir is the same phytoplasma. We found in Vineyard infected, I was erasing this, infected in Fraxinus, in Calistegia, in Robinia, in Rosa, in other species. But I want to stress that we also found group 1 in Edera helix. Edera is a kind of plants that is infesting other plants and is rarely affected by any disease. So it was able to be infected also by Aster yellows phytoplasmas. The insect vector monitoring in parallel was done, and several species, Polyphagus or also Ampelophagus, so only feeding on grapevine, were detected infected, let's say, harboring phytoplasmas. Here is a short list. Among the Ampelophagus, together with Scaphoideus titanus, we also studied Erasmoneura vulnerata. Again, we found some connection between the environment and the presence of insect vector. You see here 3 kinds of management: conventional, organic, and untreated. And you see that in the untreated one, the presence of Scaphoideus titanus and Hyalestes obsoletus or Orientus ishide is quite high. And there is a strong difference clearly seen in the graph between among the 3 different environments. In particular, for example, we found Aster yellow and stolbur or Bois Noir phytoplasma in Neoletus fenestratus. We found group 10 in Philenus spumarius. This is a question between us and the entomologist because it's not a flowing feeding, but incidentally, I detected it several times colonized by phytoplasmas. You should remember, maybe some of you know that Philenus in Italy is the vector transmitted Xylella fastidiosa in the olive region in Puglia. Here is again a list of the phytoplasma we found in insect, and what you can see in red are the phytoplasma that are not demonstrated to be transmitted yet, but found in, for example, the Hyalestes obsoletus. You have Aster yellows. In other Orientus ishide that was demonstrated to transmit flavescence dorée 5C. You can see we found other phytoplasma, group 6, 7, and others. We also found in grapevine. Here is a clear example of the same phytoplasma in the same vineyard found in both insect and grapevine. You see there is a correspondence. So the interaction between the insect and the grapevine and probably the weed, in this particular case, the weed were not reported, is quite strong and is teaching about a possible alternative cycles. So the presence of diverse phytoplasma in grapevines indicate the presence of diverse phytoplasma sources in and around the studied vineyards. So the problem can be around the vineyard, in the weeds, or in the trees, in the area around, or you can carry inside the vineyard new phytoplasma also with the propagation material, or new insects are coming and making a new cycle, a new disaster. This is nothing new. This was already, let's say, written by Antoine Caldwell 50 years ago. He was just envisaging this possibility, and it is realistic. It was completely correct. The environment plays a key role in the presence of diverse phytoplasmas and also in the presence of diverse phytoplasma strains. I didn't have the time to mention that in this survey, we also found a new strain of flavescence dorée that apparently looked very epidemic, and also of bois noir in some cases. So there is a real need not only of appropriate management for the grapevine, but also to continue to do surveys. We should not rely on what is achieved. With the epidemic disease, nothing is ever achieved, because in the time when you see quiet, it means new strains are developing, are selected by the environment, and you can have a new outbreak in the short or in the longer time. People in Veneto can teach you this, because we witnessed in the 90s a big epidemic of flavescence dorée, and now something is coming back just because they forgot about what we have set it for them 20 years ago. We just live freely, let's say without masks, and now they are again witnessing a new epidemic starting. Let's hope to keep it under control with what we learn also in the Tropicsafe. I thank you for your attention, and I hope to have saved time. If you have questions, please ask.